Hey everyone, I'm very excited to be bringing on Dr. Nathan Price. He is the Chief Scientific Officer of Thorne HealthTech, which is a publicly traded company on the NASDAQ. He is the author of The Age of Scientific Wellness. Previously, he was the CEO of Longevity, an AI health intelligence company that merged with Thorne prior to the IPO. In 2019, he was named as one of the 10 emerging leaders in health and medicine by the National Academy of Medicine, which is quite a prestigious institution. And in 2021, he was appointed to the board on life sciences of the National Academy of Sciences, Engineering, and Medicine, which is also a very prestigious institution. He spent much of his earlier career as a professor, associate director of the Institute for Systems Biology, now on leave. He's a co-director with biotechnology pioneer Leroy Hood of the Hood Price Lab for Systems Biomedicine. He is a Camille Dreyfus Teacher Scholar, received the 2016 Grace Goldsmith Award for his pioneering scientific wellness, and received a Healthy Longevity Catalyst Award from the National Academy of Medicine in 2020. Thank you very much for coming on, and we have a lot to talk about, Nathan.
Yeah, looking forward to it. Thanks so much, Joe. Glad to be here.
All right, so let's go right into it. You're the CSO of a company that sells a lot of supplements, but you have a PhD in, well, what was your PhD in again?
My PhD is in bioengineering from
Okay.
UCSD.
Okay. And then are you still a professor?
I'm an on-leave professor, so I still have some papers and still do some work on the science side. And I'm also affiliate professor at the University of Washington in bioengineering and computer science.
Okay, cool. And your area of expertise is systems biology, correct?
That is correct.
And you were also a CSO of Araval before at some
I was
point. I
one of
didn't mention that. Yeah. Which is
yeah,
a genetics company. Yeah.
was a genetics kind of big, also scientific wellness company. I was one of the co-founders with Lee Hood and Clayton Lewis, who was our CEO.
Okay. So
That was endeavor. Yeah.
you have a breadth of experience and knowledge about a whole bunch of scientific topics, especially as it relates to biology, wellness, medicine, and things like that. So let's get into supplements.
Okay.
Yeah, because you are, a leading figure in Thorne, and I wanted to know how many supplements do you take a day?
Me personally, I take probably way too many. I probably take like 20 supplements a day or something like that.
20 supplements. Okay,
A lot
all
of
right.
different.
I take more than 70. FYI.
Okay, so you're more than I. You're more than I am then. Some of those are
All right.
multi, I suppose, but
Okay. What what are let's go into what kind of supplements you take, cuz I think supplements are a bit controversial, right? There's some people, I think if you go to a regular doctor, they just say, "Okay, somebody's taking supplements, you've got expensive pee." Right? That's kind of a you know, a way of thinking of some part of America, whereas other parts are very into it. Tell me, what do you take? What are the top 10 supplements you take a day, or list as many as you can, I guess.
yeah. Although if it's okay, I'd love to do a little bit of framing about kind of why what the interest is in this kind of area, if you don't mind, in terms of kind of why I would be interested in it and whatnot as we get into this, or maybe we can do it
Well,
after.
okay, I was gonna go with the top 10 and then ask you why do you take that, but we could do that first. What are the things that you're trying to accomplish when you're either taking supplements or trying to be healthier? What are your goals?
Yeah. So I mean, so kind of the way that I come into this is really from this thing that we call scientific wellness. But basically, it's the idea that um, if you if you look broadly at sort of the kind of things that we've been doing in medicine, the foundations of medicine were really set up in the 20th century with Abraham Flexner. It was really set up for a world of chronic disease, and we had uh, a time period where and that was been incredibly successful. If you look at kind of the stuff we used to die of, we don't anymore. But if you fast forward to now and you look at chronic disease, and the way that we deal with it is typically try to find a target, drug it, that approach has not been terribly successful. So, writ large. And so if you look at that, one of the things that then drives this notion of wanting to do things early, intervene, try to extend health span, that's the whole motivation, I think, for having to do interventions that are incredibly safe, that you can do for a long period of time, and then you can deploy with some precision. And that's kind of where I came into this. Why I moved to Thorne, or why I was kind of jumped into this space. So on the supplement side, then I just want to kind of point out that natural products are just a very obvious place for us to find the kind of things that we would want to do, like in an early intervention type issue, trying to extend healthy aging, trying to do those kind of things. So from that standpoint, we do try to marry, deep testing, and AI, which are kind of my parts of the company in particular, with supplements and natural products and solutions that we're trying to do. In part because you can do, like you were saying, you take 70 supplements and I take, I don't know, 20 or 25 or something like that. But you can kind of do this like polypharmacy type thing, which you can't do in drugs. So the space is actually a lot more open for kind of a deep dive, like detailed measurements, kind of a very individualized approach per person, and in a way that on balance is generally quite safe if you're focusing on the right kinds of natural compounds. Anyway, so I just wanted to say that that's kind of where
I,
that
I
I come into,
yeah,
kind of approach.
I I wanted to go in a little more in depth in something you mentioned, which is the targeted versus broad spectrum, and that's kind of the philosophical approach to where you're coming into this, right? And that's where pharmaceuticals are very targeted and very strong in a single target, right? They're really good at getting something down or changing something or
Right.
whatever it is. Whereas natural supplements or nutraceuticals, they're more broad spectrum. And you could correct me if I'm wrong here, but that's the way I feel about it. They're more broad spectrum, which makes them safer, in my opinion, right? And also more conducive to prevention because if you have cancer already, you really sometimes need to just really target those cancer cells or target something very specific in the body. Whereas if you're just trying to do wellness and you're trying to prevent cardiovascular disease and you don't want to take a drug, cardiovascular disease could be from 10, 20, 30 reasons, right? And these natural molecules can accomplish multiple reasons. So what do you think about that
Yeah, I think
philosophy?
it's a really great point, Joe. And cancer is a great example because when you think about drugs for cancer, which are typically things like cytotoxic chemotherapy, like that's the kind of, you'll do that as a life-saving measure for late stage cancer. You would never give anything like that to a person who's otherwise well and trying to get healthier, right? Which is kind of where the more natural product approach comes in, because you are doing something that has to be essentially without side effects, and or very minimal. And so the cardiovascular is an interesting one because, as you know, there are actually compounds that you can take that will improve hemoglobin A1C, lower lipids, and so forth. Things like berberine, for example. Thorne has a new metabolic health product which is based around bergamot, which is a common natural product, I think, paired with curcumin. So there's a whole bunch of those kind of things, as you know very well, that are geared towards just a bunch of natural products that you can deploy based on blood chemistries, blood measures that you might have that you want to have it make a difference on.
Right, and something like bergamot is going to bring down lipids like LDL, but it's also going to bring down inflammation, and it has polyphenols in it that have broad spectrum activity that will do a lot of things for to prevent cardiovascular disease. And so in that regard, it's going to have usually less side effects than if you take a high dose statin and things like that. So that's a great example. I wanted to ask you about. Actually, I mean, let's bounce a little
I'd
bit
have to come
to.
back to your question about things I take or whatever. I didn't mean to
Yeah,
like.
yeah, yeah. No, no. I want to get to that for sure. That's something that I'm very curious about. That's why I just went straight to that question. I want to see where I'm coming from, who I'm dealing with, what are you taking? It's kind of like, somebody asked like what school did you go to, where do you live? For me, if I want to get to know somebody, I'm like, what supplements do you take? Just tell me already. I want to know. And then I can figure everything out about you. So, he's that type of guy. All right. All right. I know who he is.
Question.
So tell me about I love the just the general approach where you have supplements, you have testing, and you could see how these things are improving your health, right? It's not just I'm taking these supplements and then I'm guessing how these things are going to work for me. You're actually measuring, you're monitoring, and you're seeing are these things working? And obviously, people are going to try to do a whole bunch of other things like exercise and a bunch of other natural behaviors. Supplements are just that. They're supplementing the other healthy things that you're supposed to be doing.
Right.
But I just wanted to say I love that approach. Tell me a little bit about lipids, because something that very much I guess annoys me in some way is that there's a big movement to say that LDL is not really a factor in cardiovascular disease, and I feel like you would have good insights on this. And people say LDL in terms of LDLC, and there's some truth to that, but they're also saying that the LDL particle count, it's if you have the big fluffy ones, the big particles, then it doesn't matter. Tell me what your whole idea is on LDL measurement, and if you think that is a factor for cardiovascular disease.
Yeah, I'm happy to get into that. So, and I'll go down a couple avenues. So the first one is, some of the things that I think that have come out from the science community around LDLC. I think it's still, an important molecule, but there are other molecules that people have found that are, probably more important. And you brought up some of them, right? The formation or the kind of LDL particles that you have is really predictive. ApoB is actually also really important. I know a lot of doctors really feel like that's, one that they want to monitor very closely. So there are people that have really delved into that. One area that I do have some expertise on that I'm quite interested in, though, is if you're following. We mentioned you mentioned Aravel before, so we're taking people through this wellness program, and one of the things that we did monitor was LDL cholesterol. And so if people are going through a program trying to improve wellness, trying to lower LDL cholesterol, and you see mixed results in being able to do that. Now, what was really fascinating when we got in and did the data, and we published this all in a peer-reviewed paper journal called Scientific Reports. But basically, what it showed was that you could predict who would be able to lower their LDL cholesterol from their genome. And so this was, I think, the first paper, at least the first one I know of, that really showed that genome would predict the success of a lifestyle-based intervention program. So one of the things that's really fascinating is that the genome predicts a lot of the variance of the information in LDL cholesterol. So some people will have a genome prediction that predicts they'll have high LDL cholesterol and others low. So if you take people that have the same LDL cholesterol level, which would be treated exactly the same in the healthcare system today. If your genome predicts that you're high, you're very unlikely to be able to lower it other than on a statin. But if your genome predicts low and you're high, in other words, there's a gap or a delta there, then you're very likely to be able to lower it through lifestyle intervention. So by taking, and this is one of the things I'm a big believer in, because I think we should use genomic predictors across a broad swath of blood measures in the healthcare system, which we don't do today. But as we start folding the genome into healthcare, basically those deltas or those differences are highly predictive of what you can change and what you can't change, because your genome will be working with you rather than against you. So that unlocks a whole new category of variable into healthcare, which by and large doesn't exist today at all. And
That's fascinating. That's fascinating. And I have actually a specific case report myself.
oh yeah, very,
So at Self Decode, we measure one of the things that we have a genome predisposition for is LDL cholesterol, and mine actually says typical, right? And I was thinking about this, and I've tested it. The first time I tested it was 10 years ago, and it was normal 10 years ago. I think it was pretty average. It was 120, which is, I think, more or less average for the population. I wasn't on any, it was a mixed diet, omnivorous diet, not trying to bring it down or anything. And then I went on a diet that works better for me, which is kind of like a carnivore type diet, and my LDL cholesterol went to 216. And so in some way I'm looking at my report, it says typical cholesterol and it says 216. Then I said, okay, I'm going to try to bring it down now. And I started working out. I started doing things to bring it down. And by the way, I'm working against a little bit of a tide because I am eating a lot of animal foods, which is going to increase it, right? Everybody, you're on a carnivore diet, your LDL is going to go up. And I have that predisposition to go up quite high on saturated fat. Anyway, I was able to bring it down from 216 to 120 without any statins or anything like that. And so that really the reason why I'm fascinated with what you just said is because I'm looking at myself like, yeah, I brought it down 100 points, which is incredible without a statin. And it makes sense now that looking at my genetics risks and I was why I was able to bring it down quite a lot.
Yeah, and since you mentioned statins, actually, we did a paper earlier this year. This is with Sean Gibbons, longtime friend and collaborator at ISB, but basically that showed that you could actually predict the degree of efficacy of a statin, so how much a statin will affect your LDL cholesterol, and you can also predict um, side effects. I'll come to that in a second from the microbiome, right? So we have a microbiome test, right? People can take it and whatnot. But one of the things that we showed from looking at thousands of microbiome data from thousands of people was that the degree to which a statin will lower a person's LDL cholesterol, you could get a twofold difference based on the kind of microbiome that they had. And then the other big factor is if someone takes a statin, there's a 10% increase in the rate of diabetes for people that are on statins. And the likelihood, at least in the markers, of that elevating a person's hemoglobin A1C um, and or fasting glucose, so transitioning towards the diabetes, turned out that was also predictive from the microbiome. And you
Wow.
got a massive increase. So there's a certain kind of microbiome. We just split it into these different clusters. And one of those, one of the four, so about a quarter of this, um, roughly mapped into this high elevated risk for transitioning towards diabetes from the statin. So it turns out that the microbiome has much stronger predictive power for those outcomes.
I think you're unique in the sense that you have expertise in the microbiome and not just very shallow expertise. I'm talking about real expertise in the microbiome, genome, lab testing, and supplements as well, of course. But tell me how you see. I'll give you a little bit of an example of how I see them, right? Because the idea is that people want to use them all together, right? Just focusing on one very narrowly is not going to be very good, in my opinion. You need to kind of look at a lot of things. And so the way I see it is the genome genetics is really, really good in giving you predispositions for a very wide range of things, right?
Yeah.
And sometimes it could maybe prioritize some recommendations that you might want to do, or it can give you, it can give you a lot of information. But then you use the microbiome and the lab testing to track and see what your progress is like. Is that, tell me how you see it?
Yeah, I think that's one of the really most important things to look at now. So a couple of ideas. So one is you're exactly right. The genome has mostly been studied in the context of disease, right? So we have lots of these polygenic risk scores, they're called. For people that aren't familiar with that, that's essentially any particular variation in a gene, so like an A flipping to a T or a G or whatever, usually has a small effect on an outcome. But what you can do is you can sum these up. I'll give one example, which is uh, just on the science of that, which is height. So height is obviously heritable because tall parents have tall kids and short parents have short kids, and we all see that for the most part. And in the early days of this, people in genetics were confused because there was no gene for height, and there weren't like a few genes for height. You couldn't predict height from the genome. And everyone's like, we know it's heritable and you can't. But you fast forward now a decade and you can now predict height really well. It's actually the most predictable trait. But do you know how many variants it takes to predict height?
How many?
180,000. 180,000 genes that you have to sum up. So there are these things called polygenic risk scores then. So you sum up all these little effects and you can gain predictive ability. So just like you can predict height, you can also predict LDL cholesterol, which I mentioned before, and you can predict a whole bunch of other things. And most of those predictions are aimed at disease just because that's the way that our system is set up as a study. Um, uh, and one of the things we did, we published this in the Proceedings of the National Academy of Sciences a couple years ago. But we took 54 of these different polygenic risk scores, and then you can look in the blood and you can identify measures in there that are correlated with that genetic risk. So these are really interesting signals because they point you towards the earliest possible transition states, at least potentially, for that disease. Because you're talking about people that don't have any symptoms, but they just have higher risk. And I'll give you an example. So ALS, Lou Gehrig's disease, right? So ALS turned out that when we looked into the blood of people who are at high genetic risk of ALS versus not, one of the things that was unusual is that the people that were at highest risk had unusually high omega 3s. Now, the reason that that's interesting is that in general, right, usually we think omega 3 is good, right? Omega 6 is bad. And that's usually true, but not necessarily always true. And one of the interesting hypotheses that comes out of this kind of analysis is that when you is that it appears that high genetic risk for ALS is associated with having higher omega 3s. And then what was done was there was a mouse experiment that was done in a model of ALS where they actually gave the mice omega 3s. And when they did that, their symptoms of ALS accelerated. They got them younger. And when they gave them omega 6s, they got it late. They got it later in life. So that's only one axis, right? But it's fascinating because it's counterintuitive. It says, oh, if you're at that super high genetic risk, maybe you're in that top 2%, because these things tend to have kind of a hockey shape distribution of risk where it jumps up a lot at the tail. If you're in that super high risk, your trade-off for you of omega 3, omega 6s might look very different than someone in the general population. So there's all kinds of things like that, that come out of these kind of analyses that are really fascinating. We've only just starting to scratch the surface of this kind of.
Wow. You might get some carnivores hunting after you, just FYI. They're really against vegetable oils and omega 6s. But the body is very complex. In some
Generally, might
cases,
be okay, but it's there are
yeah,
details in there. They're just
right.
details. Yes.
I'm very happy you mentioned polygenic risk scoring because at Self Decode, we do use polygenic risk scoring and we're looking at many hundreds of thousands of variants. And things like any complex disease, like cardiovascular disease, atherosclerosis, diabetes, these are going to have millions of variants, or at least hundreds of thousands of variants that are contributing to this risk score. And what we're seeing on the market is a new company every day that's looking at two or three variants and telling you your prediction for some disease. Tell me
Yeah.
what you think of Tell me what you think about that. Like, can you predict any complex trait with a couple SNPs or something like that?
Yeah. So the general rule here is, and it's this one's pretty hard and fast. So if a variant, right, a gene variant, which again is just a single base pair change. So if you look at any variant that's common in the human population, right? So that it's so that some fraction of people have it that's not negligible. Those variants, pretty much by definition, have to have small effects. A 1% effect is very large in that sense. Virtually all these variants have smaller than a 1% effect. So in the early days of genomics, and some of the nutrigenomics and things like this got a bad rap early on. And part of that was exactly this. You say, oh, there's this variant and it's associated with, whatever trait. And so we're going to go coach you on it. Well, the scientific papers behind that exist, right? There's legitimately a signal, but the signal is you have like some distribution like this, and then you have another one, and it shifts by about that much, and it says, "Oh, you're in this distribution instead of that." On a personalized basis, that means almost nothing, right? It's very, very small. Um, Now, the one exception to that is that for variants that are rare, so like a rare disease in a child, for example, those can have very significant effects. In fact, Lee Hood, who's my partner in all this, and was my mentor back when I was training and all these kind of things. Like he actually published in Science the first paper on a quartet of of individuals, mother, father, and two children. And that was sufficient for them to find the gene behind this rare syndrome that was in one of the children, Miller's syndrome. And they, that was a big paper in Science. But you could do that because you're looking for a rare event. It's like a new mutation. It's something that's not going to be there in everybody, so you can dive in and actually find it. And there was also some really excellent studies Lincoln Nadal carried out at Intermountain in Utah in that hospital system, where they were looking at outcomes for kids by introducing genomes in pediatrics. And in that case, they can find these rare mutations, but they show up because they're rare. But for most of us, when you're talking about building a company where you're looking at an individual or a small set of SNPs, most of those they're not strong enough to be meaningful. But when you sum them all up into these polygenic scores, which is what we can do now, those are big enough to make a real difference. I mentioned the prediction of those for whether you could lower LDL cholesterol or whatnot. The genome signal there is strong enough for someone to be below range clinically to above range clinically. So they are, there's enough signal there that it matters. And it's growing. It's actually even a lot bigger than when we did the paper off the Aravale data. It's, I think that you can capture, I think it's twice as good now. It captured about 11% of variants then. Now it's like 20. So it's
Wow.
getting better all the time.
I'm so happy you said that, because really I think this is one of the biggest things that has caused a bad rap in genomics. Like you said, is taking a couple variants, and of course, like you said, these very rare variants are going to have a much bigger impact. But I'm talking about these variants above 1%.
For sure. They're small.
Yeah.
Yes.
Whereas, the whole consumer genomic space is basically dominated by companies taking a couple common variants and trying to predict something from it. And I keep trying to educate that you can't do that, which is why we spent many, many millions of dollars to build a system to do polygenic risk scoring, rather than, we could have taken a few scientists a couple weeks to collect a couple snips in a bunch of different okay, here's cardiovascular disease. We look on, the GWAS. What's the most significant SNPs? Here you go. But my CSO said we modeled those things, and it's basically maybe only slightly better than a coin flip, essentially, right? If
Yeah.
that.
And this is part of what I really believe in. And this is what we get into a lot in the book, The Age of Scientific Wellness, that's just about to come out, and things like that as well. But we have, if you, so what I mean when I say scientific wellness, which is, not maybe a commonly understood term, is the notion that, rather than doing all of our studies like we do now on case control of late stage disease, what we actually need to do is to study and quantify wellness as deeply as possible and generate a lot of longitudinal data for people as they're in that wellness state, earliest stage transitions, and fill in that gap, because there's very little of that kind of information. So now that we have genomic signals that actually matter, what really matters now is that we take that and then we understand how it relates to all of that ongoing, blood chemistry, our microbiomes, all the things that are going on in our bodies related to health, so that we can start to not wait until we have late debilitating symptoms that often are very hard to reverse, right? And the notion that you'll take something like Alzheimer's, the notion that you can give a single drug to an Alzheimer's patient whose half their neurons have died, and you have no chance, right? No chance.
It hasn't worked.
No,
They've been trying. Yeah.
they have, and I have a lot of ideas about. Well, anyway, that's a whole bigger topic. We've built a pretty sophisticated model of Alzheimer's over the last couple of years. But if you think about prevention, there are lots of things you can do to prevent or push off into the future the onset of mild cognitive impairment, the onset of dementia, because there's a lot of things you can do to keep your neurons alive, where you can bring it back. So that's a straightforward example. Diabetes is as well, right? Late stage diabetes, you have foot amputations and all this kind of gruesome things. Early stage prevention of diabetes is eat a healthy diet and go on a run, and maybe you wear a CGM and just learn what foods are really spiking you. And CGM is a continuous glucose monitor for those that aren't in the vernacular of that, but all those kind of things. And so that's what I think is so important, because with this idea of scientific wellness, what we want is to essentially recreate systems for health that let us stay healthier for longer, identify issues when they're small and reversible. And sometimes they can be dealt with a natural product or lifestyle or behavior, and you're not getting in, or even if a drug is particularly safe, you could, that could be appropriate. But you could, deal with these things early, be proactive, don't wait until you have this terrible problem. And the cost difference is also huge, right? Because so some of the things, like for prevention of Alzheimer's, exercise is huge because you need to maintain oxygenation into the brain. This is a huge aspect of keeping your neurons fed with energy. They also need to have enough choline. So like phosphatidylcholine, in simulations we've run, phosphatidylcholine actually becomes rate limiting as your oxygenation goes down as you get older in Alzheimer's, and it becomes harder for your neurons to stay alive. We have chemicals model simulations of this, but basically you run out of phosphatidylcholine. You can take a phosphatidylcholine supplement for your entire adult life for about $4,000 total, like for, that's across your life. Aduhelm, which was the new approved drug from the FDA that helps to clear amyloid, which, by the way, I do not believe is a cause of Alzheimer's disease. We've got
Okay.
a lot of data behind that now. Uh, and yes, I know.
Mainly, they've given drugs for it, and and it hasn't worked.
It clears amyloid, but it doesn't improve cognition. It doesn't, because it's not causal. And so when that happens, but Aduhelm, just to come back to the main point, was approved and then charged at $56,000 a year.
Wow.
So again,
Wow.
56,000 a year or 4,000 for your entire life. And people who have dietary studies, people who had diets rich in phosphatidylcholine, which is, eggs is like a primary source, they get Alzheimer's 3 years later than people who don't. So
Wow.
you're actually getting a benefit, at least from that kind of an approach, that you're not getting from, um, in a drug kind of thing. And we'll give the usual caveat of, needing, longitudinal studies and proof behind all those kind of things. But you're getting, but I think the economic argument in favor of something like scientific wellness is is very strong as the proof points really start to accumulate.
I think Alzheimer's is a great example of one of those things that start, early on, right? It doesn't start when you're 80. It starts when you're, I don't know, 30. I'm just guessing, but something like atherosclerosis, for example, I think is also a great example where it starts from the day you're born, pretty much, right? The more cholesterol, oxidized cholesterol, these things are building up in your arteries, and it's just over time it gets your arteries are getting hardened and there's calcification building up, and you don't want to prevent it when you're 80, right? I mean, it could still help if you lower your cholesterol, your ApoB and things like that, but that's something you want to prevent earlier on. And I have a polygenic risk score for atherosclerosis in self decode that puts me in the 85th percentile, which is quite significant in that, my risk is going to be significantly higher. And that means that I want to track my lab markers like ApoB most significantly. And because the higher your ApoB, that is contributing to atherosclerosis over time, even when you're 30. You're just not going to get a heart attack right away, but it's still contributing. So I really like that example. So after you started taking supplements, you've noticed improvements in your lab markers, right?
For
Is that
sure. Yeah,
yeah?
it
Okay.
revert. I was pre-diabetic. My omega threes, as I mentioned, was totally created. My vitamin D was very low. That one I do remember, because my vitamin D was, wow, it was like at 9 or 10 or something. It was like super low, and it took me a while, but up dosage until we got up into the 45-50 range that you kind of want to be in.
See, you mean to tell me that supplements aren't just making your pee more expensive?
No, I feel like well, there's been certain ones that helped me a lot in acute. So I did go through a period where I was having pretty chronic constipation for a while, which was unpleasant. That was actually my first introduction to Thorne back in the day, because someone had recommended their product just before I met any of these guys, and so I used their product, uh, Gut Mend and Fiber Mend, and took that. Cleared me right up. Was totally fine. Quit using it, went back, started it again, and then after about a month of that, then I didn't really. need to take it after that. I was pretty much resolved. And then, but some of those things I notice, or I used to have a lot of bloating, a lot of gas bloating, and I did the prebiotic plus that we've got, but which is a non-fiber based like prebiotics, polyphenol, um, all printed on this. It's actually kind of a cool thing. It's these two-dimensional printed discs that dissolve. They just make a beverage. But I do notice that, like if I have that, um, well, the cool thing there is that one of the things with probiotics is that if you just take probiotics, and I'm sure you're aware of this, they often can't compete, right? So because you've got all these existing bacteria that have been there that are really good at what they do. So when you put a new bacteria and they mostly flow through, they don't stay in particularly. So in the disc, what we do is we actually put bacteriophages in there. So it kills off some of those bad bacteria, so it creates an ecological niche, and then it creates space for the good bacteria to grow. And then you put on in there the food that they like to eat, so that they have a competitive advantage. So you can do that.
Oh,
Anyway, all that
nice.
to say is I do some of those things like for because what I'll notice is if I don't do it for a while, then I'll get like bloating or gas in my stomach, and I'll remember I used to feel that way all the time. I forget that now, but if I go back 4 or 5 years, I it was just kind of always uncomfortable. I just didn't know it was supposed to be different.
Right.
And I think there's a ton of that out there in the world where, I mean, you only live inside yourself, obviously. And so you only kind of know how you feel. And I think often we become like another thing, because I remember in, every spring in Seattle, like I would always say like seasonal affective disorder or not having sunlight never affected me. I'm like, I don't care. I'm a pretty happy person. But I'll tell you, every spring I feel like insanely happy, right? And then every spring, people talk about it, it's like every spring it comes out, you're like, wow, I'm so happy. You're like, oh, maybe it was affecting me. I just, I didn't realize it.
I
You know,
think that's a really important point that the body is really good at trying to cope with whatever state you're in and be happy with it, or else everybody would want to commit suicide. Like, oh fuck, I got all these negative things about myself. I can't stand this anymore. I think people are really good at convincing themselves I'm normal, everything's good, and there's some benefits to that. But I've noticed massive changes and like it just and through a lot of it through supplements, healthier living, and a bunch of different things. But I've noticed massive changes, and it's hard to even imagine what I was like before, because before I just thought, well, this is normal, this is how I am. It's very hard to understand how other people are. So I think that's a really good point.
yeah, and the other thing is, so we developed this biological age score, and we looked at that kind of retrospectively in the population, the data that we had collected from Arabelle. And one of the things I was surprised by is that my biological age during that 4 year period relative to my chronologic age improved by 2.7 years per year, 4 years in a row. It went down 10 years.
Wow. Wow.
And, that was partly, personalized recommendations. I got rid of my car, so I was walking to work back and forth. That was 5 miles, so that was better. I got, there was all the supplements I was doing. One thing I did, and I was a pretty big outlier for men. Women tended to do better than women got better at 1.5 year per year, men at 0.8. So I was a big outlier for the men. That's all published, by the way, in Journals of Gerontology in 2019. But when we looked at that, it was very interesting because uh, that process got better year over year. And like I said, I had been like happy and sad with sort of some of the things that had gone on. Cause like I mentioned, I lost weight, but then I kind of gained it back, and I kind of felt like I'd been up and down. But on the marker level, like for the, from the biological age, it just like ticked down, 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 down. And I was feeling a lot better. So, and one of the things I was doing, and I don't know if it's, the reason or not that I was such a big outlier, but one of the things I was doing that not everyone was doing was I was taking nicotinamide riboside, right? And this is one of, NR, and this is one of the things that's pretty interesting just from, an aging standpoint. And you mentioned some of the things that I take. I'm a big believer in, or at least a cautiously optimistic believer, let's say, in a number of these molecules, like if we can show that they extend lifespan or health span across a number of different animal systems, and they pass safety tests in humans, and you can do a functional test so you see that they're hitting the, in this case, the elevation of NAD in your cells, those are things that I'm sort of willing to take a bet on. We're still not to the point where we have long-term human studies that prove, right, that some of these things will extend health span, and especially lifespan in humans, because we live for a long time. It's going to take a while to get there. But I do think there's this whole class of molecules that are very interesting from the standpoint that they target these really conserved mechanisms. We see that they extend health span from, you know, yeast to flies to mice. And so there's at least a chance. So some of those kind of things I do incorporate into my daily routine,
Is
even in
there
the of knowing for sure that they will have that effect, but I do feel it in energy, and I take the shot that I'm hopeful that it will have the health span effect.
right? I have the same philosophy. I used to take nicotinamide riboside, and I started to take NMN.
Hmm.
Do you have any opinion on one better than the other, or just why you take NR instead of NMN? These are
Yeah,
two different forms of niacin, right? They're two different forms
yeah,
of
basically.
niacin. They increase NAD plus, just for the listener. Um, and there's kind of like a debate about what's better for aging or for health or whatever, NR or this NMN.
Yeah, my guess is that they'll have fairly similar effects because they are chemically close. Um, I have gone NR, and I'll just acknowledge I have a conflict. 'Cause, Thorne sells NR and they don't sell NMN.
Okay.
By the way, the people that are arguing the opposite have the same conflict in the opposite. So I'll just caveat with that. Um, so there's a number of reasons that we do NR though and not NMN. So for one, NR just has a lot more human data on it. NMN is sort of catching up on that front. Also, there is a debate. Both sides will claim that they're closer to the target, but I think the claim of NR is stronger, and I'll explain why. So inside the cell, NR becomes NMN, becomes NAD. So that's the argument that you hear promulgated all the time. They say, well, NR has to become NMN to become NAD. Inside the cell, that's true. But NMN cannot cross the membrane of the cell. So outside the cell, NMN has to become NR to move into the cell to become NR to become NMN again to become NAD. So since you ingest
Ah.
inside of the cell, to me, NR is closer because NMN has to be converted back in order to go across the membrane to then convert into NMN and go forward. And there's some
Mm,
complexities in the chemistry and so forth, but that's one. A second is that NR is more stable, easier to make, better, so easier from a manufacturing capability, and just and more stable, easier to ship, things of that nature. Is I think drove Thorne a lot towards that. Now, I try as best as I can to not be dogmatic on any of these things or anything if I can help it. Meaning that if really good, compelling human data came out that said, "Hey, NMN is significantly better for some reason that I don't understand currently than NR," yeah, I mean, I'll take anything that I think will extend my life, significantly. So if it comes out, but at the moment, I just feel like there's more human data for NR, and and I think the argument is stronger that it's closer to the target. Uh,
okay.
but
Let
they're probably both useful, I would imagine.
Okay, let's move to Alzheimer's. That we mentioned it a little before, and phosphatidylcholine. You said phosphatidylcholine is promising to help prevent Alzheimer's by a certain amount of time. What do you think? When I look at phosphatidylcholine, I take it, by the way,
Hmm.
but I see mixed research on phosphatidylcholine increasing TMAO, which can increase the risk of Alzheimer's. And you're an expert on the microbiome, and TMAO is this, bad thing that's produced by certain microbes in the microbiome. And when you consume phosphatidylcholine, it increases TMAO. That's the initial study said that, but then later studies said it didn't. What's your take? Does phosphatidylcholine increase TMAO? And if yes, is that a problem?
Yeah, in fact, I was going to bring it up before because I've talked about this publicly a few times, and I actually put a little bit about this into a paper I published in Nature Aging a month ago. So, yes, this is actually one of my favorite examples for personalization. So phosphatidylcholine right there. So there's some evidence that this is useful for for dementia, prevention of dementia, which we talked about. There's evidence that it's good for your liver, right, which is what I think Thorne's actual marketed claim is around. I think around just structure function related to that. Um, but the so you've got this benefit potentially that phosphatidylcholine has, but if you have, and by the way, it's not just phosphatidylcholine; it's also carnitine, right? Carnitine, phosphatidylcholine. There are certain bacteria that will eat phosphatidylcholine or carnitine and turn it into trimethylamine. Now, when trimethylamine gets into your blood, then your liver will convert that to TMAO, trimethylamine oxide, and that is associated with a number of negative outcomes. So this is why some studies, so if you have studies, for example, you go back in the older literature, they don't have the microbiome. This is why they're like, oh, it creates TMAO. Oh, no, it doesn't. Yes, it does. No, it doesn't. It depends. If there's about 20 species, we actually used to monitor them in Arabelle. So we had a test that we would give to people to monitor for them. And we just look. And we actually, and we have this, with the gut health test, like from Thorne, we can look at the same bacteria and so forth. And we're actually looking into this as part of this, what we're doing on brain health right now. But basically, that's exactly right. So you can personalize this. So in your gut, so there's a few things. One, low-level berberine. We're just got a paper on this yesterday, so just starting to read through it. But low-level berberine is inhibitory to those bacteria in the gut, makes it less likely that you'll get TMAO. So that's one thing that we're investigating right now, which is pretty interesting. The second is you do want to monitor for that. So I take a lot of phosphatidylcholine, but I monitor my gut microbiome to make sure I don't have, and currently I don't have the species that are making TMAO.
Hmm.
And I actually did a TMAO test to validate as well. And so my TMAO is not elevated. And
Okay.
so
Who sells that test?
that one I got through my doctor, so I don't recall.
Oh,
Uh,
the doctor. You could
I
get it
did
through a
that one.
doctor.
The TMAO test you can do through a doctor. I got mine
Oh,
through.
nice.
I got
Okay.
mine through a doctor on that case. In that
Okay.
case, and then of course the microbiome test. We've gotten, and probably some other people probably have. I don't know how much they do TMAO exactly, but probably they do.
And you would say the same thing for carnitine as well, whether you have those bacteria. Is it the
Yeah.
same mark?
So before I was really focused on phosphatidylcholine, that's what I primarily used it for because I like to eat red meat. But so I monitor for that. So I try to monitor my lipid levels for that, which typically have been pretty good. And then, but I did monitor for TMAO. So we had like this score that would just look at the abundance of TMAO producing molecules or I'm sorry, bacteria in the gut. And so the thing there was that I do monitor. So if it starts trending high, like I'll back off, eating
Hmm.
foods that have the carnitine. And now that I know about it, I may try um well, I'm already taking berberine. So when I saw that, I'm like, maybe that's what TMAO is like staying fine. I don't know. I gotta look more up than that. I just barely
I've
became aware of that
I've
yesterday.
got a list of 12 things that decrease TMAO. Berberine is one of the things on the list. I could send you that list,
Yeah,
um,
great. Yeah, I
or
just started
or
looking at that more.
or I could just list it off so that everybody knows about it.
Yeah.
That's probiotics, specifically L. rhamnosus GG and L. plantarum, butyrate and resistant starch, resveratrol, red yeast rice, gynostemma pentaphyllum, lycopene, pterostilbene, extra virgin olive oil, molybdenum, garlic and allicin, and celery. And for olive oil and celery, it's specific molecules in these foods. But anyway,
Oh, that's actually
yeah.
great. I take about a third of that. No wonder my TMAO is. There you go.
And,
I started taking so much phosphatidylcholine after we started doing the brain research. I was like,
exactly. Okay, that's great. We are running towards end. I know you have to go, and so yeah, tell us where people could find you. What is there anything you want to promote? Oh, you know what? We forgot one thing, if you don't mind. Just list off as many supplements as you can that you take. Go. Let's see if you can reach the 10. This is a memory test. Alzheimer's test.
Alzheimer's test. So I take our resveratrol, which is nicotinamide, riboside, and
One.
resveratrol together. I
Okay.
take quercetin. I take NAC.
Okay.
For,
NAC.
NAC for antioxidants. Let's see, vitamin D. I take the Thorne multivitamin AM and PM. Those each have 20 or 30 things in them.
Okay.
Total, let's see, berberine, Mariva,
Curcumin. Okay.
curcumin, phosphatidylcholine. Let's see, what else is in there?
Can we get to 10?
And
This is
I
a
take,
hard test, by the way.
I take glycine. I take glycine.
Glycine.
Yeah, glycine and lipids, omega. I take omega 3s.
Okay, there we go.
Let's
Bam.
see.
You got the 10.
I did 10 at least. I'll have to think
You
about
passed. You passed Joe's Alzheimer's test. The
it.
phosphatidylcholine is
Really
obviously working for you.
good.
Okay. All right. Awesome. There was a lot that we didn't get to, but unfortunately, you do have to go. And okay, so tell me where people can find you. What do you know? Is there anything you have a book coming out?
Yeah. So, main thing, if you're interested in some of the things we talked about today, The Age of Scientific Wellness is coming out. It's under the Belknap imprint of Harvard Press. It's available now on Amazon and other outlets for pre-order. I did co-write it with Lee Hood, who we mentioned a couple times. Lee, for people that don't know, invented automated DNA sequencing. It received the National Medal of Science from President Obama. Variety of many other things. One of the kind of living legends of biotechnology. So we did write this book together, and it goes through kind of this whole aspect of how we think healthcare needs to be changed around a wellness perspective, and everything else that I talked about. All the things are available at thorne.com as well.
Awesome. Thank you so much for coming here, and we shall be in touch. I appreciate it.
Great. Thanks so much. Great to talk to you.
All right. Take care. If you enjoyed this podcast, make sure to leave a review and also like and subscribe for more great content.